All right, perfect. I think we are all set up. Yeah, perfect. Here we go. All right, everyone, welcome back to Ask a Scientist. This is our first evening Ask a Scientist happy hour event, so I hope you grabbed a drink. You might need it because we're going to talk about viruses a little bit tonight. Today, I have with me Dr. Liz Anderson, who has spent a lot of her time studying immunology, the immune response to viruses. She was awarded the T32 virology grant. Yeah, and she has several publications, including some recent publications regarding COVID. So really excited to have you here this afternoon. And please just take it away.
All right, great. So I'm going to share my screen. Can you see the PowerPoint there? Perfect. All right, well, thank you. Thanks, Abby, for the introduction and for inviting me to talk. I am going to be talking about influenza pandemics in particular. And as you mentioned, immune imprinting, so the immune responses to influenza. I'm a postdoctoral researcher at the University of Pennsylvania in Philadelphia. And so I'm going to be talking a little bit about our work with the Mutter Museum in Philly. So looking for some historical specimens to sequence old flu. So just for a little bit of background, influenza has likely infected humans for millennia. It's a very old virus. However, the symptoms of flu cause respiratory disease. And so it's hard to pinpoint exactly when it started circulating. However, the first record of the word influenza can be traced back to 1504. So this was an Italian word. It meant influence of the stars. And really the reason that the Italians use this word is because nobody knew that it was a disease caused by viruses. Back in the 1500s, this idea of germs was not prevalent. So it was a disease that came in the winter, and with the cold, it was thought to have just been some, celestial force controlling this disease. And in fact, the influenza virus wasn't isolated until the 1930s. So shortly thereafter, vaccines have been developed, and now we have seasonal vaccines that are updated annually. It's also notable that there have been multiple influenza pandemics just throughout the 20th century. So of course, the 1918 flu was a huge one, and I'm going to be talking about that in more detail. But the most recent pandemic was in 2009 with the H1N1 swine flu pandemic. So as I mentioned, this is going to be a talk about influenza pandemics, but of course, we're in the middle of a different global pandemic, and it's probably on everybody's mind. I'm sure it's definitely on mine. So I wanted to give a little bit of a comparing and contrasting slide between what's going on now and what happened in 1918. So as I mentioned, the current pandemic is caused by a different virus. It's caused by coronavirus. And coronaviruses and influenza viruses are actually both enveloped viruses that have an RNA genome. So what I mean by that is that their genetic makeup or their blueprint is made of RNA instead of DNA. So there's RNA viruses and there's DNA viruses, but in particular, these are both RNA viruses. They both infect the respiratory tract and cause respiratory disease, and they're both spread through droplets or direct contact. So the current pandemic was first noticed at the end of December in 2019. And what happened was there were multiple pneumonia cases that were all linked to the seafood market in Wuhan, China. And this is a major city and a huge transportation hub. So it's very easy to understand how a local outbreak could be quickly transmitted throughout the country and the world. 1918 flu, on the other hand, was first recognized among World War I soldiers. So again, these were, it was in 1918 during the peak of the Great War, and these were soldiers that were crammed into small quarters, so a respiratory virus could spread that way. And Abby, I can't see the chat box, so if you do have any questions come up, just wave at me or something. Okay, so back to this comparing and contrasting. what's really amazing with the current pandemic is that we were able to obtain a viral genome sequence within a week. That's absolutely unprecedented, and like I'm very excited about this type of technology. On the other hand, for 1918 flu, we didn't know what influenza virus caused that pandemic until about 80 years later. So, quite a gap there. Coronavirus, the SARS-CoV-2 virus that's causing the current pandemic, is a member of Coronaviridae. So other members of that family are MERS and SARS, which has also caused pandemics. Influenza virus, on the other hand, belongs to the Orthomyxoviridae family, which contains both influenza A and B viruses. So both of these pandemics arose through a zoonotic infection, that is, an infection from an animal host that jumps into humans. We know that coronaviruses have circulated in bats for years, and there may be an intermediate host between bats and humans, but we don't know that quite yet. 1918 flu, on the other hand, and influenza viruses in general, their host reservoir are aquatic birds. So we do know that the 1918 flu came from an aquatic bird. Currently, there's over 17.7 million cases of SARS-CoV-2 worldwide, and over 680,000 deaths. Yes, you have a question.
Yeah, so we have two questions. We have one that says, have scientists tracked the rate of evolution of a virus? Are viruses mutating or evolving faster now than they have in the past?
Okay, so that's a great question, and I am going to talk about evolution, particularly with influenza viruses, and that's definitely something that we are looking at and are interested in. And we don't see that they're necessarily mutating faster than they were. So it's more of a constant evolutionary rate. But yeah, it's definitely something we're interested in.
Cool.
The other question?
Yeah, Katie asks, how do you isolate and sequence a virus in 1918? Has the technique changed over time?
Yeah, so like I said, this sequence didn't come until 80 years later. And I am also going to talk about that story because it's an incredible story. So we'll get back to that, definitely.
And lastly, we have intermediate host. Does that mean fleas or something like that?
So particularly, it's likely going to be another mammal. So pangolins were an option there for a while. That seems off the table now. But SARS was transmitted from bats to, it's called a civet cat. So likely another mammal and then a jump to humans. But yeah, great questions. All right, so the current pandemic is massive, but 1918 flu has been called the mother of all pandemics because case numbers were 500 million globally. Like that's just absolutely insane. And deaths totaled 50 million, 675,000 in the US alone. So, really big, devastating pandemics that we're talking about. I also like to show this slide because it's a map of modern travel across the globe, and it just shows how widely connected we are, and how a outbreak could occur, in one area of the globe, quickly be transmitted within that country and to other countries across the world. So, viruses don't know borders. They're just looking for susceptible hosts, and we've made it very easy for them to hitch a ride across the globe. So back to flu. Even seasonal influenza is a big deal. So not even on non-pandemic years, we have a big burden of seasonal influenza in the US even. This graph shows influenza-like illnesses over time. So seasonal flu usually peaks in winter months, with the peak being February, March, and then it decreases during the summer months. So just last year was a particularly bad season. We had, around 50 million illnesses, over 500,000 hospitalizations, and 24 to 62,000 deaths. That's, small in comparison to what's going on with COVID-19 right now. But, this is definitely something that we're concerned about every single year. And so how is flu spread? And same thing goes for coronaviruses. So as I mentioned, these are viruses that are spread through these virus-laden droplets. And there's some really disgusting videos that you can Google of people sneezing or coughing, and they've like backlit them. So you can just see like how far these droplets can travel. They can travel up to 6 feet, remain on surfaces for 24 hours. So if you touch something that has, you know, a droplet with virus in it, and then you touch your face, that's another way that you can transmit the virus. So there are some simple steps that you can take to prevent the spread of flu. And again, there's a lot of parallels with coronavirus. So, you know, unfortunately, we don't have a vaccine for coronavirus yet, but for flu, we have annual flu vaccines. So get vaccinated every year. Wash your hands with soap and water, 20 seconds at least. Wearing a mask, or, you know, following some proper cough etiquette, which is to cough into your elbow. And even better yet, stay home. Like I said before, you know, these viruses are just looking for susceptible hosts. So if we prevent that opportunity for transmission, then you can really curb these infections. So influenza disease is not just a common cold. These viruses infect epithelial cells of the respiratory tract. So I've marked here, you know, locations of cells with the flu receptor, which is different from coronavirus. Corona uses ACE2 as a receptor, whereas influenza viruses use the sialic acid receptor. Either way, it's a receptor on the host cell that the virus attaches to enter the cell. So flu infects cells of this respiratory tract and causes fever, cough, weakness, and fatigue, and can also cause pneumonia. And pneumonia can be fatal. So there's multiple mechanisms for causing pneumonia. It could be a primary viral mechanism, or the virus can ravage the lungs so badly that secondary bacteria can infect these cells and cause pneumonia. So it's definitely, you know, a big deal. So this is a, oh yeah, a question.
Yeah, we had 2 come in. So we have one that says, what is the best month to get the flu shot so it's the most effective through the season?
Yeah, so so flu shots are starting to be offered even as early as August. I typically wait until October because, as the person who asked the question is correct, your antibody response does wane over the course of the season. So, you know, you don't want to wait too late, that flu is already circulating, but yeah, October or the fall months are the best. Great question.
Is there a way to remove the type of cell that viruses are infecting?
Yeah, so with that it's difficult, right? Because those receptors and those cells serve other functions. So you can't really just remove all sialic acid because it is ubiquitous. But with other viruses such as HIV, one approach that researchers are trying is to remove the specific receptor on the cell to prevent entry. So it's definitely a good question, but it's kind of difficult when it's a cell or a receptor that serves multiple functions. Great. Okay, so so this is a depiction of what the influenza virus looks like. Again, it's an enveloped virus. It has this outer shell, and then it has these surface proteins, and we use the surface proteins subtypes to name flu. So there's different subtypes that are genetically distinct of hemagglutinin, HA, and of neuraminidase, NA, that we use for the nomenclature of flu. So for instance, H1N1 is a influenza virus that has an HA of subtype 1 and an NA of subtype 1. So that's a common circulating influenza virus, as well as H3N2. So if you've heard those terms, that's what it means. We also know of other there's like 12 HAs and multiple NAs, so there's a whole bunch. But anyway, the H1N1 and H3N2s are the main circulating viruses at the moment. And these HA spikes are necessary for replication. So this spike for influenza is analogous to the coronavirus spike. So they are these spikes that come out of the virus that attach to the cells to gain entry into the cells. So hemagglutinin is made up of two domains. You can think of it as like a head domain and a stalk domain. And when you are either infected with flu or get a vaccination, your body mounts an immune response. So you generate antibodies, and these antibodies are usually targeting this head domain because that's the domain that is used to attach to the cell. So what's a problem with flu, and is actually, luckily for us, not a big problem with coronavirus, is that influenza is constantly evolving. So in general, RNA viruses, this is back to that evolutionary rate. So RNA viruses generally have a higher mutation rate than DNA viruses. But coronavirus is really interesting in that it has it evolved a way to proofread its own genetic content to reduce that mutation rate. So this is not something that we have to worry about with coronavirus, which is great for our vaccine development efforts. But for flu is constantly evolving. So with every generation of flu, you introduce like typos into the genetic code, which can lead to mutations. And I have to say, mutations get a bad rap, but they're not always bad. Sometimes they're just, most of the time, they're harmless. But what ends up happening is that it's kind of like a game of telephone. So if you tell your neighbor, one set of sequences, they tell their neighbor, and it goes on and on and on, what you're left with is actually much different than what you started with. And for flu, this means that the evolution of the virus can actually lead to a loss in antigenicity, that is a loss in antibody binding. So the antibodies in which your body mounted in response to the teal virus no longer bind to that orange virus. And so that is the major reason that we need to update our vaccines every single year. Yes.
We have two questions coming in. We have, how many influenza viruses have been found, and why are some more common to get than others?
Yep. So as I mentioned before, there are multiple H1s, HAs, and NAs, and those can recombine. So you have a huge selection of viruses that can infect aquatic birds and mammals and humans. So there are a lot of flu viruses circulating. The most common viruses that are circulating in humans are H1N1 and H3N2, but we also have highly pathogenic strains such as H5, if you've heard of that, or H7. So those have caused outbreaks and are of concern because they're more pathogenic or they're more deadly than circulating strains. So yeah, is that.
And then we have, how do immunology researchers determine which flu will be the one for each year in order to come up with a vaccine?
Yeah, so that's a big challenge. So this is a timeline showing how vaccines are developed for flu. So in December, a year ahead of time of the season, we are constantly sequencing the flu viruses that are circulating. There are now a lot of very powerful computational models that can predict fitness or the ability for that virus to grow. And also they layer in immunity to see if immunity can force evolution into one direction or not. So it's highly complicated, and it's a reason that some years the vaccine effectiveness is not great. That being said, you can still generate cross-reactive antibodies, and they're more towards the stalk domain because that's more conserved than the head, which can protect you even if you get sick, may protect you from hospitalization or death. So you should still get your flu vaccine every year. And even if you get sick, it doesn't mean that it didn't work. It just didn't work, perfectly. But yeah, so what we do is we sequence the virus, and characterize, what's circulating, and then those viruses have to be grown up in manufacturing facilities. They have to be licensed every year by the FDA, packaged, distributed before we can do seasonal vaccinations. So this is a very long process, and again, it's not ideal for predicting something that's always evolving. So other avenues are being pursued, such as a universal influenza vaccine. And again, that's a method in which you can mount antibodies against that conserve region, the stock region. So they're hoping that you can, if you mount, effective antibodies against the stock region, it doesn't matter if the virus has evolved, you still have a robust protective immune response. So that's an exciting field that's being developed. Okay, so I mentioned this issue of antigenic drift, that is this accumulation of mutations over time. And these errors just occur during normal replication, and not all mutations are bad. But there's another mechanism that flu changes over time, and that is called antigenic shift. So in this mechanism, you have a reassortment between two viruses. So you have a human virus and an animal virus. And that can happen very quickly. And what happens is that you generate a novel virus, which humans have no prior exposure or immunity to. And this is a potential for a pandemic. So that is really something that we're constantly doing surveillance and looking out for. And so it's obvious how this pandemic potential can be transmitted to humans when there are many times that people are in very close contact with animals. So in the US, livestock shows are a big deal. Kids travel around with their livestock, and as you can see here, this girl is sleeping on her pig. So if that pig was infected, that would be a prime time to transmit across species. There's also live poultry markets. So again, the reservoir for flu, aquatic birds. So if these birds are being handled or butchered, there's a potential for transmission there. But importantly, to be a pandemic, the virus not only has to be transmitted from the animal host to a human, but also gain the ability to transmit between humans. Otherwise, if it just infects one human and then it doesn't go on to be transmitted to other humans, it just dies out. And actually, these pandemics have occurred, as I said before, throughout the 20th century. So going back to that timeline, from 1918 all the way up until 2009. And so what's really interesting is that means different subtypes have circulated throughout the 20th century. So we have H1N1 early on after 1918. There was another pandemic in the 50s that led to this H2N2 circulation that occurred until the 70s, which was replaced by H3s and then H1s and so on. And so what's interesting about this is that depending on your year of birth, you've seen different flu viruses throughout your lifetime. So an older individual would have seen this ancient H1N1 virus and H2s. A middle-aged individual only saw H2s and then the current viruses. And then children have only seen what's circulated recently. And there's something magical about childhood. So what I mean by this is that most people have actually experienced an influenza infection or vaccination by age 3. And what ends up happening is that early childhood exposure leaves a lifelong immunological imprint. So basically there are B cells, which are cells of the immune system that generate antibodies in response to an infection by a pathogen. And when a B cell recognizes the flu that you're infected with, they generate these flu-specific antibodies. And after you've resolved the infection, there's actually still a pool of memory B cells that last for your entire life. And the purpose of those is that if you're infected with that same pathogen, that they're ready to produce antibodies immediately. So it's your first line of defense with a reinfection. But what's interesting is that, you with this first infection, you get a lifelong imprint. And something that's been described with flu is that antibodies against previous strains are recalled later in life when you're reinfected. So this is a phenomenon that's been described in the 60s by Tommy Francis, and he coined this term original antigenic sin. So his idea was that someone's first exposure to influenza shapes the antibody responses that are elicited upon exposure to related strains later in life. And I've depicted that here. So if your first exposure as a child in your magical childhood was this purple virus, you mount an antibody response that wanes over time because you've cleared the infection. But then when you see another virus, that memory response is boosted. And so this can be beneficial if the antibodies can also recognize this lilac virus, but it can be detrimental if by the time you get to a white virus, you've preferentially boosted the dark purple antibodies and it can't recognize this almost white virus. So, it can go either way. It can either be protective or it can be detrimental. Yeah.
All right, so we have, will the current work on the COVID vaccines impact the ability to produce effective vaccines for the upcoming flu season?
No. So the flu season manufacturing business has been around, like I said, since the 40s. So they've got that down to a science. It is really exciting, the rapidity of the COVID-19 vaccine efforts, but I don't expect that to influence the flu vaccine at all. So, yeah.
And how confident are you that there will be an effective vaccine to combat the COVID virus?
I am confident that there will be. The question is when. And I'm hoping by next year, but like I said before, the pace at which they are rapidly doing this research is phenomenal. But in general, it usually takes years and years and years to develop a vaccine and to have it licensed and approved and everything. So it will happen, but not sure when it will happen.
Is there any way to isolate antibodies and inject them into people to introduce them?
Yes, definitely. So that is a method that we use in the lab to study protection by transfusing antibodies. This is a therapy for COVID-19 disease that you may have heard about called convalescent plasma therapy. So you take the antibodies from somebody who has recovered from COVID-19. So they presumably have, neutralizing effective antibodies in their sera or plasma, and then you transfer that into somebody who is sick, so you can help dampen the disease. So yeah, that's a great question.
I heard that the first dog died from COVID. How likely is it that my dog could be infected? And do animals typically are more immune or less immune to it?
So I haven't heard any dog antidotes. So I'm not sure about dogs. Dogs might be safe. But there have been some interesting cases with cats. And I don't know if you heard about the tiger at the Bronx Zoo that tested positive. And so it really depends on, again, that receptor that the virus uses. And felines do have this ACE2 receptor, so they may be susceptible. But research, the problem is there's so much uncertainty right now because this is so new, that we're learning things as it's evolving. So I think dogs are fine.
And lastly, do antibodies decrease for influenza over time, as we are potentially seeing with COVID?
Yes. So in general, for every infection that you get, you have this peak antibody response that wanes over time. So that is a basic immunological mechanism. Again, because your B cells are pumping out antibodies, but then once you've resolved that infection, antibodies wane. So what's happening with coronavirus and flu, after you have a vaccination or infection, you've mounted this rapid antibody response, and then that wanes over time. So I don't know if that's something to be concerned about with coronavirus. It's definitely something that happens with immunology. So, okay, so this original antigenic sin and back boosting of previous antibodies against previous strains. So our lab has shown this antibody specificity depending on year of birth in a cohort of different aged individuals who were infected with pandemic H1N1 in 2009. So this was a hospitalized cohort. So we looked at people who were born in the 50s versus 80s, and then children. And so you can imagine that somebody born in the 50s might have seen this blue virus as their first infection. Somebody in the 80s would have seen this orange virus, and an infant might not have been exposed yet. But what was interesting to see, and a proof of this original antigenic sin, is that upon infection with a pandemic H1N1, which had components of each virus, people of different ages mounted a very different antibody response. So an older individual mounted antibodies against this blue part, a middle-aged person mounted antibodies against the orange part, and then infants mounted antibodies against a different part of the virus. So they all have specified antibody repertoires dependent on their year of birth. So we wondered whether or not past influenza exposures influence clinical outcomes during that pandemic of flu in 1918. And so just to get back a little bit to this 1918 flu and the magnitude of it, again, it was the mother of all pandemics. 50 million people worldwide died. And to put that into some perspective, that's more lives that HIV has claimed in 40 years. So just really astronomical. Philadelphia, where I am based, was hit particularly hard. 12,000 people died just within 6 months. And this is kind of a timeline of 1918 flu in Philadelphia. So in July, there was already some reports from the front lines of 1918 flu. And the Philadelphia Public Health Department started to warn the public against a potential spread and a potential pandemic. In September, the Department of Health campaigned against coughing, sneezing, and spitting in public. And by September 19th, 1918 flu arrived to Philadelphia through the Navy Yard, which was a huge port in South Philly. So again, the mobilization of troops coming home and infected individuals seeded the spread of this virus. So really an ill-advised event that occurred in 1918 in Philadelphia was a Liberty Loan Parade. So this was supposed to be a parade to raise money for the wartime efforts. It was thought to be, a very patriotic parade, and we're going to do our duty to support the war. But the public health officials said, hey, there's a pandemic virus circulating. We really shouldn't have big crowds of people all together in one spot. So they advised, to cancel the parade. However, it was seen that we might look weak if we cancel it. So the parade went on, and 20,000 people lined the streets of Broad Street, which is a huge street in Philly. And unfortunately, that led to a massive transmission events. And just a few days later, over 600 new influenza cases were documented in Philly. So then the Department of Health rapidly shut down gathering places. It's kind of like what's going on now with social distancing, just close schools, close theaters, prevent spread. And a couple weeks after that was the day that the most Philadelphians died in a single day. So over 700 people died on October 15th. And this picture shows actually a photo of Broad Street. So this is the City Hall in Philadelphia. And the Spit Spreads Death was the public health campaign to kind of prevent people from spitting and coughing, and they needed something catchy. But what's really poignant is that it's pasted over a Liberty Loan parade ad. So it really shows you, you know, we should probably listen to the public health officials when it comes to epidemics. So what was really scary about 1918 flu in particular was this W-shaped curve. So typically with influenza epidemics, as shown in the dashed line, the epidemics of 1911 to 1917, so seasonal influenza, you have children and elderly dying at the highest rates. So this is a graph of specific death rate. And that's because children and elderly have weaker immune systems than healthy adults. But what was scary about 1918 is that there was actually this peak in death in otherwise healthy 25 to 35-year-olds. And what got us really interested in this peak is that if you look at the excess mortality, that is mortality over normal, again, you see it creeping up and peaking in people who were born around 1890. And what we know about 1890 is that there was a huge pandemic of flu actually in 1889. So we wonder if exposure to this pandemic led to an immunological imprint in these 25 to 35-year-olds who would have then a detrimental response once they saw 1918 later in their life. So just briefly, the 1889 pandemic, so this was another global influenza pandemic, not talked about nearly as much as 1918, but was pretty substantial. So the first reports came out in December out of St. Petersburg, Russia. And 4 months later, it spread throughout the Northern Hemisphere. Again, mobility led to the transmission, and this virus was spread by highly connected railroads. So all in all, about 1 million people died worldwide from this global pandemic. And this is a cartoon actually from the time, just saying, again, you know, kind of what we've been saying this entire talk is that the influenza is no joke. So it says, you may laugh, but it's no joke being funny with influenza. So it really can kill you. So the problem with 1889 virus is that we don't know what influenza virus caused it. Was it an H1N1? Was it an H3? Was it another subtype? That information we do not have. And the reconstruction of 1918 flu and the sequence, how we got the sequence of that is actually this incredible story that I said I'd mention later on. So this all started actually in the 1950s with a Swedish pathologist, Johan Hultin. So he had an incredible idea, and it was not mouth pipetting as he's shown doing in this picture. You really shouldn't do that, especially with infectious diseases. But his cool idea was that he wanted to study 1918 flu and understand why it was so pathogenic. And he thought, well, I need to find a specimen. I need to find an isolate that I can grow in the lab. And where can I find that isolate? So he thought about it a lot, and he remembered hearing about this Inuit village in Brevig Mission, Alaska. So this was a village in which 90% of the population was wiped out, and they were buried actually in the permafrost. And so he thought, permafrost is kind of like my freezer, and I store viruses in my freezer. I can thaw them and I can grow them in the lab and study them. So what if I went up there and got a specimen from the permafrost, brought it back to my lab, grew it, and figured out why was this pandemic so deadly? So he goes and does exactly that. He managed to get permission from the Inuit descendants to excavate their relatives. He exhumes bodies, and he brings back specimens of lungs of people who died from 1918 flu. So he gets back to his lab. I mean, this was a very long, negotiation period and excavation. He gets back to his lab and it doesn't grow. It was a big bummer. He was trying to get a PhD and it didn't even grow. But what likely happened is that permafrost, we know, has undergone multiple sessions of freezing and thawing and refreezing. And so it likely damaged the virus enough that it was no longer viable to grow in the lab. So that was a bummer. It was a great idea, a lot of effort, didn't work out. 50 years later, there's a guy named Jeff Taubenberger who worked at the Arm Institutes of Pathology. And this was in the 90s. So in the 90s, a revolutionary technique called polymerase chain reaction was invented. And this allowed us to take genetic material and amplify it up so that we could sequence it. So this has changed the course of molecular biology. So Jeff Taubenberger was working at the Arm Institutes, and they had formalin fixed tissue from soldiers from 1918. So these are, very thinly sliced lung tissue that are on slides for pathologists to understand the pathology and understand why people died. And they had access to these samples. So he tries to sequence 1918 flu from this type of specimen, but it doesn't work. So it's likely that there was just not enough. And then a 72-year-old Johann, he hears about this. And so he contacts Taubenberger. He knows, like, this specimen was probably too small. You need something bigger, and I know where to get it. So he goes back to Alaska. He gets permission again. He tells them, I know 50 years ago it didn't work out, but this time we have this new technique. It's going to happen. So they let him dig up exhumed bodies again. He gets specimens, ships them to Taubenberger and Ann Reed, and they're able to sequence the virus. And that's where we found out that it's an H1N1, and every segment of the virus was originally from an avian source. So the moral of this story is really that technology is amazing and perseverance pays off. That's 50-year story of how we got a sequence. So that's cool. We have 1918 flu sequence, but we still don't have the 1889 sequence. And our question is, will imprinting with this 1889 flu leave 25 to 35-year-olds susceptible and ill-equipped to fight 1918 flu? So in order to understand that, we really need to reconstruct the 1889 pandemic virus. And so I'm in a pretty cool spot at the University of Pennsylvania and have some opportunities to look for old specimens that we could potentially recover 1889 flu from. So I just want to give a little bit of overview of where I am. So University of Pennsylvania was founded by Benjamin Franklin in 1740. The medical school that I am at is the first medical school in the country. It's a medical school that was founded before our country was a country. And we also have these elite groups of physicians called the College of Physicians. So they were instrumental in curbing the yellow fever epidemic in the 1700s. They also led to the development of the Mutter Museum and all of the really interesting specimens that we have there. And it wasn't until the late 1800s that the medical school moved from downtown Philadelphia out to Western Philly, and they built a hospital. So the idea was for medical students to actually get some hands-on learning before they were just released into practicing. It's a great idea. But what's cool about that is that we can look for flu samples from these medical students starting from the late 1800s. And this lines up perfectly with, the time frame of what we're looking for. We're looking for what flu circulated between 1889 and 1918. And so just to start with the endeavors that I've gone through, looking for these specimens, we found autopsy books from the 1800s, and these are absolutely incredible. So again, the stories of specimen hunting, there's always some adventure. So these autopsy books were found wrapped in brown paper bags, like stuffed in a closet somewhere. Somebody opened them up and said, "Huh, that looks important." So they brought them up, and we've actually had them digitalized and restored. So this is a picture of what it looked like before restoration. It's completely falling apart. But what's cool about these is that they're handwritten documents of autopsies that were performed by the medical school. And so we've identified multiple cases that death was by pneumonia. Again, in the 1800s, like germ theory was starting to emerge, but viruses, it was still iffy, and flu wasn't known yet. So I really was excited to see this entry because they actually called it influenza. So we're hoping to match up this specimen soon. Unfortunately, the location of the specimens are probably also likely stuffed into a closet somewhere. And so those are elusive as of yet. What we have found is another avenue, eBay. So you can't make this stuff up. I was perusing eBay, and we found lung tissue slides from a different medical school in Philadelphia. So Jefferson Medical College. It's also a very old, renowned medical school in Philly. And it's likely that this was a student's personal set, but they had in particular lung tissue. So I bought them all up. It was kind of creepy. I asked the seller, where did you get these? Can you tell me more information? He got like creeped out by that and just stopped talking to me. But he did send them. So we don't really have any history on them, but we had our pathologist take a look, and he was really excited that there were a couple very promising specimens for us to sequence from. So what are the next steps? We are planning to extract the viral RNA, again, the genome of influenza from these specimens. We can amplify that up and sequence it. And now we have techniques, unlike what Taubenberger was dealing with in the 90s, we have these next generation deep sequencing techniques. So again, technology is always evolving. And now we can sequence very small amounts of genetic material. We can then take those sequences and map them to the influenza genome and see what subtype caused the 1889 pandemic. And so just to kind of summarize, and what I hope we will learn when we do this, whenever our labs reopen, is what influenza subtypes circulated prior to 1918, and whether or not sequential infection with 1889 flu and then followed by 1918 flu led to a focused antibody response. So as is so important, when we look at the past, we can prepare for the future. Understanding past pandemics will allow us to better prepare for the inevitable, the next influenza pandemic. And with that, I'd like to thank you all for your attention and for joining us. Thank you, Abby, for inviting me. My mentor, Scott Hensley, and everybody in his lab, our other collaborators at the University of Pennsylvania, our collaborators outside of the University of Pennsylvania, especially our folks at the Mutter Museum that are helping me track down some other specimens there. And then I also know that this is a time of a lot of confusion and misinformation. So I wanted to put up some resources that I value and trust for you to check out for the current pandemic and also for influenza information. So yeah, can take any questions.
Yeah, so I hope you can hear me. It's thundering and lightning and raining, but I'm
no.
on my porch. So we have a couple questions left. We have, do you know if the 1918 pandemic had the same type of political implications that we've seen with COVID-19? 200,000 people attend a parade in Philly despite official health warnings. That's crazy.
Yeah, no, it's really interesting to kind of compare and contrast the political climate back then versus now. So again, it was in the middle of a war. So that having that parade, was supposed to be like to support the war, very patriotic, but it totally backfired. Yeah, it's absolutely crazy. Another though interesting thing with the political climate was mask wearing. So once it got real and once people understood the magnitude of the pandemic, wearing a mask was seen as a patriotic act because you were helping to curb the epidemic that was affecting our soldiers and our economy. So that's an interesting kind of difference that I see now.
Yeah, absolutely. And then we'll finish up with this last question that says, can you speak to herd immunity?
Yeah, so that's a great question. So herd immunity is it's this mechanism by which you have enough of the susceptible population exposed to a virus, for instance, that they mount an antibody response, and so that's another way to curb an epidemic or a pandemic. However, with the current coronavirus pandemic, we are nowhere near herd immunity. So it's definitely not a good idea to try to stop this pandemic by herd immunity. The other way you can gain herd immunity, though, is through vaccination. So that is what we need to wait for. But yeah.
Oh, I would like you to speak just real quick about what would you say to people that are a little reluctant or nervous to get vaccines?
Yeah, so getting that timeline that I showed you of vaccination for developing and licensing an influenza vaccine. So that's like a year-long process, and that's for a platform that has been, well vetted, used over and over and over. So getting to a safe vaccine takes a lot of work. And what's amazing right now is that we're doing the work at a rapid speed. But I wouldn't be reluctant. I understand why people are, but the thing is, there's so much safety and evaluations that go into this that I wouldn't be concerned.
All right. Well, thank you so much, Dr. Liz, for your expert opinions on all of this and all of this information. It's really valuable, especially at a time like this. We had a couple people chime in and say, wow, this is extremely relevant. So
good. Yeah.
Yeah, good times for it. Thank you so much for spending your evening with us. And thank you everybody for who tuned in and send in your questions. I would love to hear feedback, whether people prefer this segment to be at night or versus the day. So let me know, comment from me too. But thank you so much, Liz. We'll talk to you soon.
Thanks.
Bye.